scientists were aware that there must be some kind of genetic material that was carrying information from, say, parent to child, or from one cell to another as they divide. And one of the reasons why is we had done some interesting experiments that demonstrated there must be some sort of physical information carrier. Now, we didn't know what it was at first. We thought it was probably proteins. Proteins do everything else interesting inside of the cell. We thought probably not DNA because, well, you know, it's just A, T, C, and G. Just 4 bases there. How could 4 bases be complex enough to code for something as wonderfully complicated as a human being, or an oak tree, or a dog, or whatever it is that the DNA would have to be coding for? Now, proteins, there's 20 different amino acids in proteins. Surely that is enough complexity to allow a cell to code for a human being, or an Escherichia coli bacteria, or anything of that sort. Now, there was some experiments done by a guy named Griffith that demonstrated that there must be some sort of chemical material which was carrying information for traits. And he demonstrated this with an experiment with bacteria and lab mice. So he took 2 different strains of bacteria. They're related, but they are slightly different. They have different traits. One is rough and the other is smooth. The rough strain is safe. You can inject it into a mouse and they will live, and that's what he did. He proved that it would live if it got the rough bacteria. The smooth is virulent. It is dangerous. It will kill the mouse if it's injected into it. And he injected into some lab mice in order to demonstrate that it would do that, that it had that trait. Then he did something else. He heat killed the smooth strain and he injected into the mouse. Now, why did he do that? To prove that the material of the smooth strain by itself was not enough to kill the mouse. There wasn't just a toxin inside there that was killing the mouse. It had to be live cells, live bacteria that were being injected in order to kill the mouse. So he heat killed it. Heat killed means we literally took a flame and we killed the bacteria. He took that heat killed bacteria and he injected it into the mouse. Mouse lives as though nothing happened because the bacteria is dead. So even though it was the deadly strain, it's no longer able to do any kind of damage. Then he did something interesting. He took the heat-killed smooth strain bacteria and he took the live rough bacteria. Now, again, look at this slide here. Rough, by itself, didn't do any damage to the mouse. It lived when it got injected with the rough strain. That was the safe strain. The heat-killed smooth strain bacteria didn't do any damage to the mouse. It was fine by itself. But when he mixed the 2 together and he gave it to the mouse, well, what happened? The mouse died. And then they checked, and some of the bacteria that were inside that mouse were now of the smooth strain. Somehow, information for having that smooth strain virulent trait, being a deadly bacteria as a trait, was transferred to the rough strain bacteria that we injected in. So there must be some sort of physical material that was left behind from the smooth bacteria in order to allow that transformation. This is what it's called. When bacteria pick up DNA from their environment and they use that to alter their own traits, we call it bacterial transformation. So there must be something. We thought maybe it's proteins, maybe it's DNA, but there's something that's carrying information for these traits. Now, this idea of bacterial transformation was researched a step further by another group of scientists. They took what Griffith had discovered and they made the next logical step. These scientists were named Avery and MacLeod, and what they did was they got enzymes that would break apart specific types of molecules. So they got an RNase, something that would break down specifically RNA. They got a DNase, something that would break down DNA. They got a lipase that would break down lipids. They got a protease, an enzyme that breaks down proteins. A carboase, which is an enzyme that breaks down carbohydrates, right? So logical enough. They took a flask of the smooth strain bacteria and they heat killed it. Then they poured that heat killed smooth strain deadly bacteria, formerly deadly, now dead, into 5 different flasks. To each flask, they added 1 of these special enzymes. So they were getting rid of all of the RNA in this flask. They were getting rid of all of the protein in the second flask. They were getting rid of all of the DNA in the third flask, and so on and so forth. And then they mixed in the live rough strain bacteria. In every single case, they got deadly smooth strain bacteria in the final check. When they checked their flasks at the end, they got smooth strain bacteria, except for the flask in which the DNA had been destroyed. So suddenly, we have a paradigm shift, right? We suddenly realize it wasn't the protein that was the information carrier, it's probably the DNA. But one experiment is never enough to completely change the state of science. Let's do a confirmation. So then this brings us to Hershey and Chase. Hershey and Chase do some experiments on viruses, a particular kind of virus called a bacteriophage. Bacteriophages, bacteria, and phage, eater, they attack bacteria. Bacteriophage attacks bacteria. Now, viruses have only two kinds of molecules in their structure. They have protein and they have DNA. What Hershey and Chase wanted to find out is what material was the virus actually injecting into the bacteria. Now, in order to understand this, you need to understand how viruses reproduce. A virus will attack some other cell, some living cell, and it will inject its genetic material into the machinery, the reproductive machinery of that cell. So it will try to get some of its DNA into your DNA, so that when your cell goes and copies itself or it copies its DNA, it will also copy the virus DNA, and it'll make more viruses as a result. They're kind of a parasitic reproducer. So what Hershey and Chase wanted to know was, is the protein in the virus being injected in, is that the material that's being copied and used to make more viruses, or is the DNA being injected? In order to determine this, they used radioactive elements. They used radioactive isotopes. Proteins contain sulfur. There are certain amino acids, cysteine is an example, which have sulfur in their structure. So if you grow viruses in radioactive sulfur, they're going to use that sulfur in their proteins, and all of the protein will be radioactively tagged as a result. DNA does not contain any sulfur, so only the protein would be radioactive and the DNA would not be radioactive. Another group of viruses, they grew in radioactive phosphorus. There is no phosphorus inside of proteins. None of the amino acids use phosphorus, but DNA does use phosphorus. So when they grew those viruses, you had radioactive DNA, but non-radioactive proteins. We have two different kinds of viruses here. One's where we have a radioactive tag on the protein, another where we have a radioactive tag on the DNA. What's going to happen? So this virus comes in and attacks Escherichia coli, E. coli, a normal bacteria that we use, and it's probably the most common lab bacterium that we use. What they found was that the protein was left outside of the bacteria. Only the DNA was actually being injected inside. Now, viruses are not living organisms, and they caution, we shouldn't jump to conclusions based on this result. But with this plus Griffith's results, this is what really caused the paradigm shift. Everyone now realizes, oh my God, we need to get on this DNA thing. We need to figure out what DNA looks like, and that's where you got Watson and Crick and Wilkins and Franklin coming in and trying to solve that problem. Now, I mentioned this next scientist already, but let's get his name on a slide, Chargaff. Chargaff found out that for any amount of adenine that you have in a sample of DNA, you get an equal amount of thymine. For any amount of cytosine that you have, you get an equal amount of guanine. So you can figure out how much of every single nucleotide base is in a sample of DNA if you just know one, if you know how much one is valued inside that sample. And this clued Watson and Crick into the base pairing rules, and that's what it's called, base pairing. On one strand of DNA, if it reads TCAG, on the other strand, it's going to read AGTC, going straight down that line. A to T and C to G. Easy mnemonic, angular letters with angular letters. A to T. Look at all those angles in those letters. A to T. Curvy letters with curvy letters. C to G. C to G. Whenever you see a G, you put a C. Whenever you see a C, there's a G on the other side. Now, there is another nucleic acid, and that is RNA. And RNA doesn't quite play by the same rules. C and G are always paired together, right? Whenever you have G, guaranteed there's C on the other side. Whenever you have G, guaranteed there's C on the other side. But RNA is a single-stranded nucleotide, and it doesn't have any thymine in it. There's no T. So whenever you would have a T, you'll put a U instead. I'll give you some practical examples where that becomes a lot more clear, but if you see any U's in a polynucleotide strand, it's definitely RNA. If you see any T's, it's definitely DNA.